Hello.
Morning. Well, here we are again, gentlemen. Cameron, good morning. You know, whenever I go to the school district website, Cameron, I see your recognition, and you know, it's been so weird being in the classroom and out of the classroom all at the same time. I wanted to offer my congratulations on that award. It's a pretty cool accomplishment, man.
Thank you.
Thanks for logging into the Pear Deck slide. We'll get started here in just a minute. Thanks for logging into the Pear Deck slide. Good morning. Trying to remember how to get everything set up all at the same time. There's a bunch of folks. People are still joining us. Get everything muted here. All right. We got just about everybody. Good morning, Tiny Faces. Once again, or white names on a black background. Once again. I hope you're doing okay. That's the transition. Plan for the day as we come back to a remote learning stance. I do have a check-in for you. It's new and old all at the same time. Uses a tool that we've used in the past, but hopefully uses it in a new way. And hopefully it'll give us a chance to ground ourselves, but also to remember, especially this time of year, if you're celebrating the holiday on Thursday, that it's an opportunity to be grateful and maybe even to direct those feelings of gratitude towards others, whether they are family members, loved ones, strangers, friends, it doesn't really matter. So it's a brief practice that I'm hoping you're willing to play around with a bit here at the beginning. I hope you had a chance to watch the EdPulse video on the biological functional groups. I have some practice problems that we can use to hopefully to develop a deeper understanding of those groups, and then eventually begin to look at, okay, great, you've got these functional groups, they're part of larger biological molecules. As back to last week, right, we know that we should be able to put molecules together, we should be able to take molecules apart, and there are specific types of reactions that are responsible for doing that. So if we think about anabolic processes, there are reactions in biology called dehydration reactions that put molecules together. There are also catabolic or hydrolysis reactions that take molecules apart. They're similar to each other, but of course accomplish different goals. And my hope is that we can look at some of those, but also use some practice questions to develop a deeper understanding. And we'll wrap up things with an exit slip like we try to do each and every day. And then actually one other thing that wasn't on the agenda that I thought might be helpful, and I'll give you a quick poll before we even get started with our check-in here. There were some extra ATP mediated energy transfer questions that were part of the slide deck. I encourage you to take a look at them last week, but I'm wondering, would it be helpful today to do one of those together before we go on to the new material? If you could just drag your dot quickly to give me a sense of whether that would be useful to you or not. Okay, looks like that's going to be helpful for most people, so we can definitely do that. Okay. All right then. Thank you for doing that. I've got a question ready to go, or a couple questions ready to go that we can use for that. Hold this, whoops, sorry. So if you're willing, just try something a little bit different. This practice is a practice that's been really important to me as I engage in it. It's called loving kindness. And really, in shorthand, it's a way to think about happiness, something that, I don't know, I'm in dire need of right now in my life. Maybe you are too. But happiness is contagious. Have you ever heard the expression, what is it, 7 degrees of separation or 5 degrees of Kevin Bacon? Right? As human beings, we are interconnected with other human beings. And what this practice is all about is that we can bring to mind somebody who's close to us, or even somebody that we don't know very well, and we can direct positive thoughts, we can direct happiness towards them, while simultaneously reaping the benefits of sending that happiness out into the world. We can actually make ourselves happier as we push happiness out and direct it towards others, whether they're close to us or not. So this is a brief practice, and I'm hoping you're willing to play around a bit. And it begins actually with a tool that we've used in the past. It's our mood meter. But in the past, we've used the mood meter really to direct or really for me to see how you're doing. This morning, what I was hoping you'd be willing to do is to drag the heart to the mood meter, to a place where as you bring someone that you care about, someone that you're close to into your mind, how would you like that person to feel today? Okay. So dragging your heart to the spot on the mood meter that identifies how you'd like somebody else to feel. A lot of people have already responded. Thank you for doing that. All right. So what I'd like you to do is to bring that person to mind, whoever it is. In fact, it doesn't even need to be a person. It could be a pet. Someone that you're close to, someone that you'd like to direct positive feelings towards. But really bring this person to mind. Get a sense of what they look like, what they smell like. And try to imagine this person doing the things that make them smile, that make them happiest. You don't need to think too hard about this. It can simply be the first thing that comes to mind as you think of that person or that pet. As you bring this person to mind and you picture what it is that they're doing that makes them happiest, take a moment to notice how that makes you feel. You can even take your hand and put it on your heart if you'd like. So we're going to send some kind wishes, some kind thoughts to this person or thing or pet. And it's really simple. We're going to use an internal voice as we direct these thoughts, these feelings towards the other person, towards the other thing. And as I say the statement, you can simply repeat it in your own head. Okay, but try to keep that person in your mind as you do this. And notice how it makes you feel as you do it as well. Okay? I wish for you to be healthy. I wish for you to be happy. I wish for you to be peaceful. I wish for you to have joy in your life. And as we wrap up this very simple practice, try to imagine how that person or that pet is feeling as you send those positive wishes, those positive thoughts towards them. And you can take a deep breath. And it's as simple as that. Curious to see what you were wishing for other people. You can wish these things for yourself, of course, as well. I sent a message out yesterday. There's no homework over the break for this course. I'm hoping that you'll take the time to be with the people that you care about most, who love you the most, who you love the most, wherever that is, however that is, and that you'll take care of yourselves. My hope is that other teachers are doing the same, that they're not bombarding you with essays, projects, tests upon the return to school next week, and getting, once again, this overwhelming sense that you folks are overwhelmed. You deserve a break. You're worthy of a break. And I want to make sure that I can do whatever I can to give that to you. So that's it. Thanks for sharing. If you played around with a bit with heartfulness, with loving kindness, I appreciate it. And it sounds like from that earlier poll, you would like to do a practice question around the ATP-mediated energy transfer. So this is a sample free response question from a released AP exam. This class may have done one similar to this, whether you were in the AM or the PM section, but different classes got to different points last week. So this is a new question regardless, but it might look similar to one that you did. Take a moment to read the question, to study the figure, ask any clarifying questions if you have them, and then to begin writing a response. See, some people have started writing, that's great. feel like you're getting stuck, can you identify the catabolic reaction, the exergonic reaction, since we're focusing on the energy releasing reaction in this question? Can you identify the anabolic reaction, the endergonic reaction, the one that's absorbing energy in this process? And if you can identify those reactions, can you describe how the energy releasing reaction is working? Remember, heat energy, thermal energy, is not useful to living systems. So we're trying to move away from this exothermic or endothermic processes, and again, think of them as exergonic or endergonic processes, right? They're referring to the free energy that's being made available to do work, rather than heat energy, which as a living thing, we can't do anything with directly. Maybe think about the models that we played with last week, the simple models that tried to show you phosphate group transfer and how energy was being transferred between ATP and other biological molecules. I'm seeing a lot of people have answered this question. How much more time, if any, do you need to wrap up writing your response? You can show me with your fingers if you're on camera, or post something in the chat if you can't. For a finger, 1 minute, fist is 0. I'm seeing a lot of fists from people on camera. Appreciate that. If you can't turn your camera on, I totally understand it, but if you could put something in the chat just so I know, either you're ready to move forward or you still need some more time to finish. Thanks, Vivian. Thanks. Okay. Thanks, Annabelle. Would it be helpful if I kind of took you through the answer to this question? Okay. There is a scoring guide, and I can show you the scoring guide. It's a pretty simple scoring guide in this case. And again, this is the College Board's language, not my language. It simply states the response includes a description that a phosphate group is removed from the molecule of ATP and that ATP is converted into ADP, right? So how might you actually compose, right, an answer that addresses those points? Well, the first thing that we want to make sure that we're recognizing, as I pull my pen up here, just for a moment, is that there's two reactions that are taking place here, right? There's the catabolic breakdown of ATP, which we have shown here. And we have the anabolic synthesis of this molecule, which you've never seen before. You don't have to have seen it before to answer this question. Fructose 1,6-bisphosphate, which is abbreviated FBP for short. Okay. So what are we doing in this reaction? Well, we're breaking down the ATP into ADP, and we're transferring that third high energy phosphate group from ATP to this molecule of fructose 6-phosphate, or F6P. Okay. So we're taking the energy that was present in that third phosphate group of ATP, and we're transferring it to fructose 6-phosphate to synthesize, to create a molecule of fructose 1,6-bisphosphate. Okay. So in a problem like this, if we go back to the models that we were playing with last week, fructose 6-phosphate is biomolecule X. We're breaking down the ATP, we're catabolizing the ATP, and we're transferring a phosphate group to molecule X in such a way that it is now energized, right? And while we don't see it in an illustration like this, because it's 2 dimensions, we would also expect that the shape of fructose 6-phosphate would have changed as a result of being energized upon the addition of that phosphate group, okay? So really question just popped up. When we see on the diagram where the phosphate group comes from, well, I think that stoichiometrically, we would say that if this is ATP and this is ADP, we've lost a phosphate group. Originally, fructose 6-phosphate had one phosphate group, right? What we've done is added a phosphate group to fructose 6-phosphate at that position. Okay, that's the first carbon for what it's worth. So that's going to form fructose 1,6-bisphosphate. So really what we've done, again, is we transferred the phosphate group from ATP to fructose 6-phosphate to make a molecule of fructose 1,6-bisphosphate. That's the anabolic process. And we achieved that by breaking down or catabolizing ATP and transferring the energy from ATP, but really the third phosphate group of ATP, directly to fructose 6-phosphate to make fructose 1,6-bisphosphate. Yeah, this is a skeleton structure, and I'm never quite sure exactly how familiar you are from chemistry with skeleton structures. The CH2 is still there. I'll change colors to make it a little bit clearer. This little point of intersection, that's a carbon atom. It's just not shown. And what also isn't shown because it's a skeleton structure is that there would be 2 hydrogen atoms that are attached to that carbon atom. So you can kind of picture one here and one there. You know, that carbon is sp3 hybridized. It's got to have a tetra array of atoms around it or bonds around it. All of those rules that you learned in chemistry, right? Not just memorized for the test, are in play. So the CH2 is still there. And this is actually one of the examples of the reactions that we're going to be looking at today. The addition of that phosphate group was achieved by dehydration synthesis. And what we ended up doing, as we'll see in a little bit, is we've lost a molecule of water. So the OH that was part of that group was lost to the surroundings in the form of a water molecule. If you're curious about where the CH2OH went, well, that's where the OH went. The CH2 is still part of that larger molecule. Does that help?
I'm just a little confused. Like, where's the ATP in the original, I guess, I don't know what to call it, diagram? Because there's only one phosphate group, but isn't that triphosphate? Like, where's it coming from?
Right.
Like, you just not draw ATP?
Yeah, so it's almost like a shorthand way. The molecule of ATP has not been drawn, right? Nor has ADP, nor has the phosphate group for that matter. But the catabolic reaction is depicted in what I tried to highlight in yellow, while the anabolic endergonic process is what's depicted in more detail up above. I guess not up above, from left to right would be a better way to say it.
Okay.
So there's 2 reactions that are taking place, right? We're trying to move energy from a catabolic process into an anabolic process. So both reactions are there. right? It's just that one is depicted more thoroughly, right? The anabolic reaction is depicted more thoroughly in more detail, while the catabolic process, the breakdown of ATP, is just shown in like shorthand.
Okay, that makes sense.
Trying to make sure I don't miss anything in the chat. I think I got everything. Okay, does that help? Thumbs up if that helps. Okay. It's tricky. It's tricky, right? What I think is most important is that we're trying to illustrate the movement of energy from one molecule to another molecule. If ATP is really acting as the energy shuttle, right, and that energy shuttling is being accomplished through phosphate group transfer, right, we can go back to the model that we played with last week in class, or if you did it virtually from home, it's okay too. Or we can look at a reaction like this and see that we're still accomplishing the same goal. We're moving a phosphate group and its associated energy from ATP to some other biological molecule as it's being synthesized anabolically, endergonically. Okay. Is it okay if I erase all this? All right. Well, what I'm noticing as I look at all of the released items that the College Board is making available to teachers is that they're asking the same question in like 3 different ways quite often. So this question looks identical to the one that you just answered, but I'm trying to highlight the part of the question that's different. Okay. So I would encourage you, if you feel like you need, if you're kind of in that area of, I'm not quite sure what's happening here, keep practicing, right? And I'm including the scoring guides so that you can look and see what your answer needs to include. Of course, I'm always more than willing to take a look at a draft of an answer that you've composed and give you some feedback if that also would be helpful, okay? This is how we get better, right? We practice, we get feedback, we revise if necessary. We don't cram the night before an exam, right? That's not learning. It might promote success in some cases, and it might have even promoted success for you in the past, but that's not learning. Our brains need opportunity to practice in order to gain a deeper understanding of what's happening. So I would encourage you to keep practicing if you feel like that would be useful to you. So sorry for that previous question. So if I'm describing the energy releasing reaction shown in figure 1, I would say something to the effect of the breakdown of ATP and the release of the 3rd high energy phosphate group leads to the transfer of energy from ATP to fructose 6-phosphate as fructose 1,6 bisphosphate is being synthesized. That's how I would answer that question, in my own words, of course.
Would you categorize that as anabolic because the molecule needs energy to absorb to make that bond?
So the fructose 6-phosphate to fructose 1,6 bisphosphate is absolutely the anabolic reaction. The catabolic reaction is the breakdown of ATP.
Yeah, okay, sounds good.
Any other questions about that before we move forward? Okay. Well, then as we move forward, my hope is you had a chance to watch the Edpuzzle video on functional groups. Because what we're trying to do, right, is we're trying to continue to develop a deeper understanding of, catabolism and anabolism. It's all perfectly well and good to, have a general understanding that catabolism, breaks down biological molecules, anabolism builds or synthesizes biological molecules. But in this next part, when we look at functional groups, it's going to be the functional groups themselves that are reacting to either build molecules anabolically, right, so that the new molecule will have more energy than the reactants do. Or if we're catabolizing biological molecules, it will still be those functional groups that are reacting to form new molecules as the original molecule is broken down catabolically. Okay. So in the video, you were introduced to this chart, and you better memorize this chart if you want to be successful. in this course. He says sarcastically, right? You know, if you're going to continue in biology, is it useful to know these functional groups? Absolutely, because you're going to see them again and again and again and again and again. They're never going to go away. But would it, if it's helpful to you to like print off that and have it with you in your notes, or to come back to it electronically whenever you need it, then do that, right? This isn't about memorizing. This is about applying. So what I'm hoping as you watch that video is that you, as I try to introduce each of the functional groups, and really the ones in the video are the most important functional groups. There are some on this chart that you may encounter, but really are rare in the context of biological molecules. What I'm hoping you took away from the video is that not only are these functional groups the parts of the molecules that are going to react, they're also the parts of the molecules that instill chemical properties onto the molecules, right? So, you know, hydroxyl groups are polar. So if you had a biological molecule with a lot of hydroxyl groups, chances are that molecule is going to be polar in nature, and so on and so forth. So I'm hoping that that video that I made, you know, in my own voice, was helpful to you. But just pausing there before I give you some practice problems to dive into, do you have any general, like, surface questions about functional groups in biology?
So are they used to transfer the phosphate group to the molecule?
Sometimes, yep. Yeah, so sometimes you can almost think of them as, especially hydroxyl groups, that's going to be a really common place where the phosphate group will be added to a biological molecule. And trying to think, we're going to see the carboxyl group a lot when we look at protein biochemistry, as will the amino group. That'll be a really popular functional group in protein biochemistry. And we'll look at, you know, how changes to those functional groups actually lead to different structural changes in the shape of a protein. Yeah, but the others often can be thought of, especially the hydroxyl group, as spots where phosphate groups could be added for sure. Okay. Well, let's do this. I have some practice questions for you on functional groups. Let's see, they're on slides 89, some people already started, 90, 91, 92, okay? So what I'd like to do is give you control of the slides, answer these functional group questions to the best of your ability, but also give yourself a chance to take a short break. So, sorry, I'm just gonna give you student mode. Okay, so let's say probably 4, maybe 5 minutes tops to answer the questions, and then we'll take a short break. So we'll come back together right around 9:40, 9:39, 9:40. But please, after you answer the questions, or before you answer the questions, give yourself an opportunity to stand up, stretch, get a drink of water, use the restroom, take care of yourselves. You can stop when you get to slide 92. You don't have to answer the one on slide 93, mostly because we haven't done that yet. the lesson that's referenced at least. So 89, 90, 91, 92. Since you're in student mode too, if you want to go back to that chart, you can always do that. Use it as a reference. I guess you can take a screenshot too, right? If you don't want to click. If you're not going to take a break or you already took your break, I'd love to know what you think about using Pear Deck in this way. You could unmute yourself, pop something on the chat. Obviously, I've never tried this before. So just in terms of identifying something on an image, did you find this useful? Did you not find it useful? I'd love to hear your feedback if you've got any. Thank you, Will. A thumbs up is good feedback. I like that. Thanks, Trevor. Thanks, Jesse. I'm glad you enjoy dragging dots. All right, this is a good time to come back together. It's about 9:39. I hope you had a chance to take a break. All righty. Well, let's see here. So obviously in this first one, as we look at your responses, there's slightly different ways you can answer the question, especially for the hydroxyl group, because there's more than one there, right? And you only had one blue dot to drag. But hopefully you're identifying the part of the molecule that has the OH, right? So it's typically, I mean, in biology, is a carbon atom that's then attached to an OH group. And there's several different OH groups that are part of this molecule. It looks like the aldehyde group was also identified correctly by most people. The aldehyde in this case, and really in all cases, it's going to be present at the end of a biological molecule, because if I come out of this so we can actually see the molecule, so it's not covered by your dots. Not that your dots aren't beautiful, just that it's a little hard to see with all those dots covering that part of the molecule. The aldehyde functional group, it's one of the two carbonyl functional groups, is C double bond OH. So C double bond OH. Okay. So things look good there. You identified the hydroxyl group and the aldehyde carbonyl functional group. If you're wondering, this molecule is fructose, one of the simple sugars that often we ingest as part of our diets, especially now in a first world nation where high fructose corn syrup is so common. Whoops, need my mouse. So things look good there. Well done. Going on to the next one, the carboxyl functional group. Well, this one, hopefully, very nice. It's at the end of the molecule as well. It's a C double bond O, OH functional group. Often instills acidic chemical properties. We'll be looking at that later on, but I'm hoping you took that a little bit, took that away from the video as you watched it last night or the night before. Well done there. Moving through this. Again, blue for the hydroxyl functional group, and then red for the ketone, the other carbonyl functional group. Where you drag your blue dot might vary because there are several hydroxyl groups that are part of this molecule. This is actually glucose, the molecule that is catabolized in cell respiration. But it looks like for the most part, we have identified the ketone functional group correctly. But as I switch between the two views, you're seeing what I'm seeing, right? You're seeing your dots and then you're not seeing your dots. Okay, good, thank you. So the ketone functional group is sitting right here. So it's still a C double bond O, so that's why it's a carbonyl functional group. But rather than being attached to a carbon and then a hydrogen atom, the ketone functional groups can often sit in the middle of two carbon atoms. So it's, in this case, it's attached to two other carbon atoms. So well done there. And I think there's just one more. I get rid of the ink. Blue for the amino, yellow, orange for the carboxyl, and then red for the hydroxyl. Looks good. Oh, some dots are still dragging. So blue for the amino is the NH2 group, very nice. And then the carboxyl group in orange, C double bond O, OH. Looks like some people dragged their red dot to the OH that's part of the carboxyl. You want to be careful there. In that case, the entire functional group is the carboxyl functional group. Even though there's an OH there, we wouldn't classify it as a hydroxyl group because it's part of a larger functional group. But there is an OH down here at the bottom of the molecule. This is actually a molecule called tyrosine, one of the 20 amino acids that make up proteins. So ideally, you would have dragged the red dot to the bottom of this molecule where the hydroxyl group is located. Okay. So a quick overview of the important biological functional groups. Is this bringing up any questions for you on your end before we go forward?
I had a question about the previous slide.
Sure. Come off of it.
So, okay, so we're looking at the ketone. I'm looking at the ketone functional group, and it's a oxygen atom double bonded to a carbon, and then that carbon's also bonded to two other carbons, but that carbon at the top is also connected to the H2OH. So I was wondering if that still like counts as part of the ketone group, because it could be a larger.
It's a good question. I think you're kind of thinking about this part or what else is attached to this carbon. And really it could be anything. It doesn't really matter. So no, it would not specifically alter our ability to identify this, what's in the circle, as the ketone functional group. Specifically, it would be the C double bond O, and then the 2 carbons that are attached. So if I were to change colors one more time, the 2 carbons that are attached to this carbon that are going to make up the ketone functional group, whatever else is attached to these 2 carbons. That's a terrible way of writing that. So carbon 1 and carbon 2 is inconsequential.
Okay. So for the next slide, then we had that hydroxyl group that wasn't really a hydroxyl group because it was connected to the carbon double bonded O. So that's like a larger group, but that doesn't apply to the previous slide.
Well, no, I mean, this is not a carbonyl group, right? It's not an aldehyde group, it's not a ketone group, because in order for that to be true, once again, if I pull up my pen here, yes, we have a C double bond O, right? And you might argue, okay, well, that's a carbonyl group, but it's not an aldehyde because it's not attached to a carbon and then a hydrogen. So it's not something that looks, if I were to sketch it really quickly, something that looks like this. Okay, that would be an aldehyde. And it's also not a ketone because to be a ketone functional group, we'd have to have something that looks like this. We don't have that either. Right? We have C double bond O for sure, but then we have this OH that's attached to the carbon. And that's what makes this entire, I'm running out of space, entire functional group a carboxyl group and not an aldehyde or a ketone group.
It's also not a hydroxyl group, right?
No, the OH is not, I mean, I understand where you're going, right? Because there is an OH there and it looks like a hydroxyl group because it's attached to the C double bond O. All together, what I'm trying to draw in the circle here would be a carboxyl group and not a hydroxyl group or even a carbonyl group in this case, based on what the other atoms or how the other atoms are arranged relative to one another.
Okay.
Okay. So again, this isn't about memorizing, right? This is about identifying. Go ahead, Trevor.
So there could be a, no, never mind. Never mind.
Okay. If something comes up, let me know. All right. So with this in mind, again, our goal is to think about these functional groups in terms of the parts of the molecules that are reacting, either to form biological molecules anabolically, endogonically, or these are going to be the parts of the molecules that are going to react as biological molecules are taken apart, catabolically or exergonically. Okay. So I move ahead here a little bit. There's all sorts of reactions that are taking place inside of a typical cell, right? Some of these reactions are anabolic. Some of these reactions are catabolic. So another way to earn an A+ in this class is to memorize this illustration and to redraw it to scale with the correct color scheme on our next assessment. That's preposterous, right? But this is what, all these are the reactions that are taking place inside of a typical plant cell, right? So you can't memorize this stuff, right? It's ridiculous to try to memorize this stuff. It's way too complex. So what we're going to try to do is use these functional groups, right, to think about how molecules are being put together in cells like the ones that you're looking at right now, but also how molecules are being taken apart in cells like you're looking at right now without memorizing the reaction, because that just seems like an incredibly futile task given the complexity of the systems that we're working with. But you can do it if you want to. Alrighty. So here's what's great about biology. When we put together molecules, when we take apart molecules, more often than not, it's like putting together a puzzle with small pieces coming together to make a larger whole, or we're taking apart that puzzle into smaller pieces as we make a more complex molecule less complex. So you might have even encountered these terms in chemistry before. In biology, we're going to assemble molecules from monomers, from single molecules. So as we combine them together, we're going to make more complex molecules anabolically endergonically. They're going to have more energy than their constituents do. And as we assemble those monomers together, what we're going to begin to build are polymers, much larger molecules that are built up from their individual monomers. And this is going to be true whether we're building sugars like carbohydrates, proteins, fats, or even nucleic acids like DNA or RNA. They're all built up from monomers, from smaller subunits. And what's super useful in biology is that if we're going to build a biological molecule by assembling it from monomers, we're going to use the same type of chemical reaction again and again and again. Okay? In the notes down below, I refer to this reaction as a condensation reaction. The College Board often uses the term dehydration reaction instead of condensation. So just a quick note, they mean exactly the same. Okay? Condensation reactions and dehydration reactions mean exactly the same. And they're really simple. If I have a reaction, so I pull up my pen here once again, where I'm trying to put two monomers together, or in this example, I've got a short polymer, and I'm trying to add a single monomer to that short polymer. Notice I've got a hydroxyl functional group, and in this really simple reaction, a hydrogen atom on the other molecule, the other short polymer. And if I'm going to add that monomer to the short polymer, this reaction is going to allow for the monomer to become covalently bound to that short polymer. So here's our new covalent bond that's forming. And a product of this reaction all of the time is a single molecule of water. Where's the water coming from? It's coming from the OH, the hydroxyl group on the monomer, and in this example, the hydrogen from the short polymer, OH2, H2O, there's where the water is coming from. This is actually why they're called condensation or dehydration reactions, because we're taking a molecule out, right? A molecule of water out as a product. Thus, we're dehydrating the product as that new bond forms between the constituents. Okay. Does that bring up any questions for you? So I'm going to try a couple practice questions before we wrap up. Well then, this is an AP practice question. It's a multiple choice question. Two molecules, you probably don't recognize them, but you might recognize their functional groups. Which answer makes the most sense to you? Hoping you're seeing what I'm seeing. So two monomers up top, we're linking them together. We can see the molecule of water that's being produced. Looks like a lot of people have answered. Let's see how you did. Yeah. Right? The name of the bond isn't super relevant right now, but as your answers change quickly. This is an example of a dehydration or a condensation reaction. Two monomers coming together, a new covalent bond is forming between the monomers, and a molecule of water is produced along the way. So it's an anabolic endergonic reaction. Right? We're synthesizing molecules. That product is going to have more chemical potential energy associated with it than either of the two reactants do on their own. Right? Maybe you even noticed the functional groups that were involved in the formation of that bond. So as we wrap up here, which functional group is that? Not everybody at once.
Carboxyl.
The carboxyl group. And then over here, which functional group is that?
Amino.
The amino. So where's the water coming from in a reaction like this? The OH of the carboxyl group and one of the hydrogen atoms from the amino group are combining to form water, and a new covalent bond forms between the two monomers. It's a dehydration reaction. We're synthesizing a new molecule from two monomers. Okay? All right. So we have just about reached the end of our time together. And I don't know that I'm going to be able to find the exit slip fast enough, but we'll see if I can really quickly. If not, yeah, we didn't even get to the hydrolysis reactions. So if you do have a moment just before you sign off, in a minute or less, what's your most important takeaway from the lesson today? Whatever it is. It might be a single sentence. As soon as you've added your reflection, you're welcome to sign off. Please take care of yourselves for this next week. I won't see you for a week. Do well. Give the people that you love a hug. Take care of yourselves, for sure. I hope that you will.
Thank you. Bye.
Thanks.
Thank you.
Thank you. Bye.
You, Mr. Bergeron.
Take care, everybody.
Thank you.
You're welcome.
Thank you.
Good morning, Sarah. Ben and Max, good morning.
Good morning. Hello.
Hello, hello. It's weird. I want to say hi as soon as you come in, and then it says connecting to audio, and it doesn't make sense for me to say hi until here come a bunch of people. Here we are again, together but far away. Keep logging into those Pear Deck slides. We're going to use them a lot today. But then when don't we? That's not what I want. Alrighty. Well, good morning, everybody. Welcome, wherever you are. Our plan for today, I have our usual check-in, but this one's different, although it uses a tool that we've used before. And I can tell you a little bit more about it here in just a moment. It's a way that maybe we can address exactly what's going to happen this week in terms of our family get-togethers and our loved ones and who are most important to us right now. So I thought it might be useful this time of year. I actually do have a, I mentioned this last time, but with the AM PM schedule last week and even the week before, I found it all to be very disconcerting. So actually, let me do this before we even get to the check-in. I had and have ATP mediated energy transfer free response practice questions ready to go. So I'm just wondering, do you think trying to answer one of those questions and then going over it together as a group would be helpful for you this morning? If you could just drag your dot really quickly. It gives me a sense of whether or not this would be helpful or if you just like to move forward with the new material for today. Let's see where you're, oops. Okay, that's fair. That's kind of what I figured. All right, so after our brief check-in here, we can hop into one of those ATP mediated energy transfer questions. Again, they're the same questions that I linked to from the last class, but if you didn't get a chance to look at those over the weekend, they weren't homework, of course, they're just there for you to practice. I thought using a few minutes here today would be helpful for you. So we will do that. So if I go back to the agenda here, then we want to spend some time focusing on what I'm hoping you were introduced to in the Edpuzzle video on biological functional groups, and take some time to practice identifying functional groups, not because you're ever going to have to memorize them, but because I want you to be able to, if you even had that chart that was part of the video in front of you, could you use that chart to identify functional groups? And then thinking about how those functional groups are actually useful to us as we connect, catabolism and anabolism and functional groups together to either build biological molecules anabolically, endergonically, or take apart biological molecules catabolically or exergonically. Because believe it or not, it will be those functional groups that are reacting to form the molecules or to cause the molecules to break apart as a result of their chemistry, really of their structures. That's really what it comes down to. So I'm hoping you're kind of seeing the story that's unfurling over these last couple of weeks. All right. Well, then, if that's our plan, before we get started, do you have any questions for me or for the group that we need to address? Okay. Well, our practice, our check-in for today, it actually involves a sub-practice of mindfulness that's referred to as loving kindness, sometimes referred to as heartfulness. And really what it's all about is bringing an attention, bringing awareness to the fact that we actually have the ability as human beings to influence, albeit indirectly, because we can't control the emotional outcomes of another human being, but that we can influence how much happier a person, either really close to us or even really far away from us in terms of our connection to them, feels over the course of the day. Ultimately, as we send these positive, loving, kind thoughts outward, what it gives us a chance to do is to notice how that makes us feel. Because while we may not be able to influence the emotional state of another, we certainly have control over our emotional state. So as we practice this really simple skill today, I would invite you to notice how it's making you feel rather than how it's making somebody else feel, because you really can't know how somebody else feels unless they tell you. So the practice begins really simply. What I'd like you to do is to bring someone to mind. It could be someone close to you. It might not be someone close to you at all. It could be someone that you walked by yesterday and that you don't know very well. It could even be a pet for that matter. It doesn't have to be a human. But I'd like you to bring that person to mind. Really picture them. And with the heart, if you could wish for that person or that pet to feel a certain way, drag your heart to the box, to the word, to the color that best represents how you want that person to feel. I know it's abstract. Bring a person to mind. Really picture them. Maybe even picture them doing something that makes them happy, that brings joy to their life. And send a wish to them in the form of dragging your heart to the appropriate word or color or box. So again, these practices are really simple. With that same person in mind, again, picturing them. If it helps to close your eyes, you're welcome to do that, but you certainly don't have to. Imagine them doing something that makes them happiest, that brings joy to them, that makes them smile. And it can be the first image. We're not going for any sort of perfection here. If you bring that person to mind and you picture them doing something that brings joy to their life, notice how that makes you feel. Maybe even put your hand on your heart. If you've never done this before, be kind to yourself. This is hard. So what we're going to do is, we already did it a little bit with our heart dragging, but now we're going to send positive wishes, positive intents, positive thoughts towards that person or that pet on purpose. So I'm going to read some really simple phrases. You can repeat them as you picture that person in your mind. And just imagine that you're sending those wishes outward to that person or to that pet. Maybe even allow yourself to imagine how it makes that person feel as they receive them from you. Okay. I wish for you to be healthy. I wish for you to be happy. I wish for you to be free from suffering. I wish for you to have joy in your life. Try to imagine how sending that out into the world makes that person feel. Maybe it makes them happier and happier as they receive your wishes. But also notice how it makes you feel to send those wishes to that person on purpose. And that's it. It's that time of year, right? If you're celebrating the holiday on Thursday, if you're going to be with your family, if you're going to be away from your family, it's an opportunity, right? To send these wishes to them, especially because you may not be able to be with them in person this year. So I would encourage you to play around with this practice, right? You've got paying attention to sound and paying attention to your breath and even sending positive thoughts out into the world. So thank you for practicing with me. On to the biology. All right, so you voted overwhelmingly for a practice question. And what I've noticed is the College Board has been releasing items to teachers where they've taken a question and they've really written 3 different forms of that same question. So I don't remember if this class got an opportunity to tackle one of these practice questions, but even if you did, this one is different. Okay, so unless you practice on your own, this question should be totally unfamiliar to you. So take a moment to read the question, to study the diagram, ask any clarifying questions that you might have either out loud or in the chat, and then do your best to answer this question in the box that you have on the right-hand side. Perhaps I'm seeing a lot of people haven't had a chance to write, either with fingers if you can be on camera, but if you can't, just in the chat. How much more time, if any, do you need to finish composing your answer? Thank you, thank you. Lots of zeros. Okay. Okay. So, since you've finished, would it be helpful to go over this answer together? Okay, lots of yeses are popping up. Okay, all the thumbs up. Thank you for that. It helps me. Okay, well, let me first show you the scoring guide for this question. Again, this is a scoring guide not written by me. It's written by the College Board, and then we can talk about how their answer applies to this particular question. So their answer simply says the response includes a description that a phosphate group is removed from the molecule of ATP and that ATP is converted to ADP. So it's pretty generic, right? It may not even be very helpful to you, but it's what the answer would need to include in order to gain credit. And honestly, for a question like this entire answer is worth 1 point on the AP exam, okay? So let's go back and kind of unpack this a little bit. So here's a reaction that you've never seen before, and that's going to happen on the AP exam. Heck, it happens in our own assessments, right? There's a scenario, a problem, a reaction that you've never seen before, but can you apply your understanding to solve the problem? So in a question like this, as I bring my pen up, we really have two reactions that are taking place, right? One of them is the catabolism of ATP. ATP is being broken down into ADP and the phosphate group. So if we went back to the model that we were playing with last week with those 3 styrofoam balls, right? This is like removing that third styrofoam ball, removing that third high energy phosphate group from ATP. What are you left with? Well, a molecule of ADP. So this is your catabolic reaction. This is your exergonic reaction. We use the term that we've been using for these last few class meetings, okay? But there's another reaction that's taking place, right? Because we're trying to transfer the energy from the exergonic process to a process that we know absorbs energy, right? The endergonic or the anabolic process. Well, that process is depicted, if I were to change color, it's going to be kind of tricky here, with all of the structures that are shown in the diagram, okay? So a molecule of fructose 6-phosphate, F6P, is having a phosphate group added to it to synthesize a molecule of fructose 1,6-bisphosphate, or FBP for short, okay? So if this is one of our reactants, okay, the phosphate group, the high energy phosphate group that was part of ATP broke off and is being transferred. Here, let's do this. Change colors one more time. So if this is that high energy phosphate group, I know there's 3 of them, right? Then that high energy phosphate group has been transferred to fructose 6-phosphate to synthesize a molecule of fructose 1,6-bisphosphate, okay? So the energy that was part of ATP, right, has been transferred, at least partially, through phosphate group transfer to fructose 6-phosphate to make a molecule which now has more chemical potential energy, fructose 1,6-bisphosphate, FBP for short, right, as a product of the reaction. We could even, think about that third question that we were working on last week, where there's an exergonic process that's associated with an anabolic process. Change colors. So maybe something like I don't know, my penmanship isn't great this morning, right? Or the energy that's coming out of the exergonic process, so in this case, ATP catabolism, right? That's the orange line, is being directed towards the anabolic endergonic process, the red line. Some of that energy, of course, is going to escape to the surroundings in the form of heat. We can never avoid that loss of thermal energy to the surroundings, which is actually a good thing because it makes sure that our second law of thermodynamics is not being violated. But as a consequence, the products, right, are going to have more energy than the reactants did because the phosphate group and its associated energy has been transferred from one molecule, ATP, to fructose 6-phosphate, even draw it in here, to make a molecule of fructose 1,6-bisphosphate. So the product has more energy than the reactant did because it's accepted a phosphate group from ATP. So let me just pause there. What questions is this bringing up, if anything at all?
What are they expecting our answer to look like? Do we need to include all of this or?
It's always hard to say, right? Because the College Board is being very general in terms of their scoring guide. So what I would write, right? If I go back to the scoring guide, I'm going to leave the ink up, so I apologize if it's a distraction. I would say, since my response needs to include a description that a phosphate group is removed from the molecule of ATP and that ATP is converted to ADP, right? What I would say is that, in my own words, right, the catabolism of ATP leads to the release of a high energy phosphate group. The high energy phosphate group is transferred from ATP to fructose 6-phosphate, F6P, to make a molecule of fructose bisphosphate, FBP. Done. Right? Something like that. Because the energy is coming from one molecule, right, really being transferred from one molecule, that'd be a better way to say it. And it's being transferred through that phosphate group, right? The phosphate group is binding to F6P, and it's making a molecule of FBP that has more energy associated with it. Okay, so again, something like the catabolism of ATP releases a high energy phosphate group. The phosphate group transfers energy from fructose 6-phosphate to synthesize a molecule of fructose 1,6-bisphosphate that has more energy associated with it. Something like that. What other questions does this bring into light for you? Is it okay if I erase this? So there are other questions that are part of our slide deck that you can practice with. Again, the College Board is creating a question and then really just generating 3 different versions of that question. So same process, right? Same setup, if you will. But this time the question is asking for the transfer of energy that occurs in the reaction. Sorry, no, that's what we just did, right? No, sorry, no. So this is a little bit more specific. This is the transfer of energy that occurs in the reaction in figure 1. So this might even be getting to the whole piece of the puzzle, right? Energy is released from ATP, captured in the formation of the new bond between F6P and the phosphate to form FBP, right? Or energy is transferred from the ATP molecule to the F6 molecule to form FBP. That's kind of the whole process of energy transfer, where it looks like if we just focus on the energy releasing reaction, right, that's where the ATP is being catabolized and the energy is being released as that phosphate group leaves the molecule and ends up being transferred to fructose 6-phosphate to make fructose bisphosphate. So specifically, we could probably answer just this question by addressing the catabolism of ATP, wherein this other practice question, we're building in the answer that I just described that actually includes the energy transfer from ATP and a corresponding increase of energy, right? The input of energy, the absorption of energy as fructose 6-phosphate is converted into fructose 1,6-bisphosphate, right? So there's multiple opportunities to practice here, and that's how we get better, right? We don't cram the night before an exam. That's not learning. That's not understanding. It might work, which probably means the tests are fatally flawed. Right? But it's not how our brains learn. Our brains learn as we practice, as we get feedback, as we go back and revise our answers to make them that much better. So as you continue to learn, my hope is that you'll look at these practice problems and use them to your advantage to develop a deeper understanding, not because they're homework, not because they're worth points, not because they improve your grade, but that they improve your understanding. Nah, why bother? All right. Is it okay to move forward? Well then, the video that I'm hoping you watched for today, the Edpuzzle video, introduced you to, I think it's 5 of the most important biological functional groups that we're going to see as we continue to explore biochemistry. And again, as we connect this to what we've been learning about, right, if there are catabolic reactions that break down molecules and there are anabolic reactions that build biological molecules, where we're trying to go is, okay, great. Well, if I'm putting molecules together, if I'm building a molecule, what parts of the molecule are reacting, right, that allow those molecules to come together? Well, those are going to be functional groups. And similarly, if I have a biological molecule and I'm breaking it down catabolically, well, what parts of the molecule are going to be reacting to break that molecule apart? Well, those are going to be biological functional groups. And while I highlighted the 5 most important in the video, there are others, okay? So you need to memorize all of these in order to be successful in life. Of course you don't. You know, this electronic copy, I suppose you could take a screenshot if you wanted to. I'm sure you could print this off if you wanted to. It's a resource, right? This isn't about memorizing, it's about understanding, okay? So what I want to do is give you an opportunity, if you'd like to use this chart, you certainly can, to identify some of the different functional groups that are part of biological molecules, okay? So here's what I was thinking. I've got, I think, 4 slides of questions. So starting on slide 89, 90, 91, and 92 have opportunities for you to drag a dot to some of the different functional groups you were introduced to in the Edpuzzle video. So it's 10:30 now. Why don't we do this? Why don't we take maybe 7-8 minutes tops to do the dragging, keeping in mind that you can always go back to the chart if you need a refresher on which functional group is which, but also give yourself an opportunity to take a break along the way. So 2 or 3 minute break as well. And then we will reconvene at 10:38. I will give you control of the slides so that you can drag your dots at your own pace. And of course, I'll be here if you have questions along the way. And because you're in student mode, you can go back to the chart as well. and take that break. We'll come back together in about 2 minutes. Stand up, get a drink of water. Stretch, mindfully yawn. Same for me. Alrighty. Shall we come back together? Is anybody out there? Thanks, Mac. So just some practice with identifying the different functional groups. Again, why are we doing this? Because it's going to be these functional groups, these parts of biological molecules that are going to be reacting to either form larger biological molecules, those are our anabolic processes, or these are going to be the parts of larger biological molecules that are going to be reacting as those molecules break apart catabolically to make smaller biological molecules. So I'm going to take you out of student mode here, just so that we can look at things together. So my hope is you identified one, there's several different hydroxyl groups that are part of this molecule. If you're wondering, this is fructose, good old high fructose corn syrup, a very common ingredient in processed foods in first world countries and other countries for that matter. So things look good there. And the aldehyde functional group looks like there may be a little bit of uncertainty. So let's just take a look at that briefly before we go too much further. The aldehyde functional group is one of the two carbonyl functional groups. So there's going to be a C double bond O, that's part of its structure. But what makes an aldehyde group different from its cousin, the ketone functional group, is that that C double bond O will be attached to a hydrogen atom, like this. Which means that the aldehyde functional group is always going to be found at the end of a biological molecule, never anywhere near the middle or really anywhere other than the end for that matter. So this is what I was hoping you identify as the aldehyde functional group. Again, there are several different hydroxyl groups that you may have identified. So that looks good. The carboxyl functional group, beautiful. C double bond OH. Okay, it's not a carbonyl group. It kind of looks like a carbonyl group, but it's not a carbonyl group because it's not bound to, the C double bond O is not bound to a hydrogen, like in the aldehyde group. And it's not a ketone functional group because this C double bond O here is not bound to two other carbon atoms. Stay tuned, that's our next question. It's bound to this OH, okay? It also means that this OH is not a hydroxyl functional group because it's attached to this C double bond O. It's actually where the name comes from, carboxyl. Carbonyl, C double bond O, hydroxyl OH, carboxyl. Okay. So that looks good. Moving to the next one. And if you have questions, please feel free to ask either out loud or in the chat. I want to know what you're thinking. I want to know if you're having any trouble. Again, now we're identifying a hydroxyl group, but now a ketone functional group. So let's see how you did. Lots of different choices for our blue. Some dots are still moving around. I love that. People correcting or revising their answers. So lots of different places to identify a hydroxyl group. No one right answer there. It looks like everyone now is targeting that red ketone functional group. And what makes it a ketone functional group? Well, again, C double bond, oops, sorry. C double bond O, but now I've got, rather than a hydrogen at one end, I've got that C double bond, that carbon that's attached to the oxygen via the double bond, attached to two other carbon atoms instead. That makes it a ketone functional group. This is actually glucose. This is the molecule that is being catabolized in cell respiration, like we saw last week. Alrighty. And then the last one, I tried to create one that had a couple more functional groups associated with it. So amino, carboxyl, and yet another hydroxyl. How'd you do? Oops, sorry. The blue dot is the amino group, beautiful, looks good there. And then the carboxyl group is up top, that's the orange. Again, be careful, if you're dragging the red dot to that OH part of the carboxyl, that's not a hydroxyl group. I know it looks like one, right? But because it's attached to that C that's double bound to the oxygen, the whole group is the carboxyl group. And then down here at the bottom of the molecule where you just have the OH, that's the hydroxyl group in this example. I love that people change their answers, that's great. There's actually something in our brains that registers the fact that you're dragging that dot to the correct answer as a way to solidify your understanding of a concept like this, because this isn't so much a concept as it is an identification. All right, so just some different examples there. Before we move on to look at how these functional groups are actually reacting, what questions do you have? Silence falls over the crowd. All right. So there are lots and lots and lots of different biological reactions that are taking place inside of a single cell. This image is a plant cell. So a third way to get an instant A+ in this class, remember human dissection without anesthesia has to be you, unfortunately. We have to sterilize you in the autoclave here in the classroom. Or you can memorize this image and redraw it on our next assessment day, take-home assessment day. So you're encouraged to do that if you would find that to be useful to you. My gosh, please don't do that. That's crazy. Right? This is meant to show you that you can't memorize this stuff, right? It's too complex. It's too complex. Instead, what we're going to try to do is to identify trends that exist whenever molecules are being put together or whenever biological molecules are being taken apart, because all of that is happening in a diagram like this as you look at it. Alrighty. So how are these molecules being put together or taken apart? The great thing about biology is that the reactions that we're going to use to either put biological molecules together or take biological molecules apart really only come in two different forms. And I think we're going to have time to address the first form today. We'll have to say the second reaction, really the breakdown reaction for next time, next week. So if we're going to build biological molecules, so again, we're talking about anabolic processes, we're talking about endergonic processes, we're talking about processes that are going to absorb energy. We're often going to do that by assembling monomers together. You can think of monomers as just single molecules. Okay, so if we had two monomers and we link them together, we'd have a short polymer, a larger molecule that is composed of, in that example, two monomers. Right? Simple as that. And you know, a polymer really refers to a large biological molecule. If we only linked 2 monomers together, we'd really only have a dimer. If we linked 3 together, we'd have a trimer. If we linked 4 together, we'd have a tetramer, so on and so forth. But for a course like this, we can just call any molecule that is composed of 2 or more monomers as a polymer, okay? Well, how do we assemble these polymers, whether they're short or long, from their monomeric constituents? We use a type of reaction referred to as either a condensation reaction, or the College Board likes to use the term dehydration reaction. The 2 terms are synonymous. They are entirely interchangeable, okay? So in the example on the lower right, we've got a short polymer. It consists of 3 monomers that have already been linked together through condensation, through dehydration reactions. And what we're going to do is link 1 more monomer to that existing polymer, all right? So how do we do it? Well, in a simple example like this, well, looky here. We've got a hydroxyl group. And looky here in this simple example, we've got a hydrogen atom. What's going to happen as this new monomer joins this short polymer is we're going to form a new covalent bond between the existing polymer and that new monomer, so everything gets linked together. And the reason why we refer to these reactions as either condensation or dehydration is that we're getting a molecule of water out as a product. In fact, this is why the functional groups matter. Because in this example, it's the functional groups that are leading to the production of that water molecule. OH plus H, OH2, H2O, that's the molecule of water. Comes out, enters the surroundings, a new chemical bond forms between the polymer and that single monomer. Okay. So shall we try to practice a little bit with this? Thank you, Alex. It's nice to see somebody responding. Thank you. It really helps me to know that you're out there. This is tough for teachers too. I hope you know that. This is really tough for teachers. Even if they're saying otherwise, trust me, this is really hard for teachers. All right, so a couple different practice questions that we can look at before we wrap up today. First one is an AP multiple choice question. Okay, so you don't know what these molecules are. You don't have to. See if you can choose the answer that makes the most sense. Responses coming in fast and furiously. All right, try to choose the answer that makes the most sense to you. We'll take a look at how you did. Beautiful. This is the formation, or showing the formation, of a covalent bond. Yes, it is a peptide bond, but you don't know that yet, and in many ways it's not relevant to answering this question. And it is a dehydration synthesis reaction. Right? We're putting 2 monomers together to make a very short polymer, a dimer, if you will. Okay. Thank you for changing your answers too. Again, why or how does this connect to our functional group understanding? Well, take a look. Here's your amino group. Here's your carboxyl group. These are the functional groups that are going to react, in this case, to form a new chemical bond. It is a covalent bond. Yes, it's a peptide bond. That's coming, okay? And as a consequence, since this is a dehydration reaction, or if you prefer, condensation reaction, water is a product. Well, where's the water coming from? Well, if I were to change colors, the OH from the carboxyl group is combining with one of the H's from the amino group. That's where the water is coming from. And as a result of the reaction, an anabolic, endergonic, energy-absorbing process, we make a larger molecule from the two monomers. Okay. Want to try one more? We have time for one more. All right, this one's a little bit different. Now you're going to enter a number as your answer. Oh, sorry, that should be, since it's of a polysaccharide, not the protein. Sorry. It's not going to change your answer. But that's
what are monosaccharides?
Monosaccharides would be the monomers that are going to be used to build the polysaccharide, which is the polymer in this case. Sorry, the protein part, that should be polysaccharide. That's my mistake. Different way of thinking about it. If we took two monomers and we linked them together, we get out a molecule of water. If we linked 25 monomers together, how many molecules of water would we produce as a product? Getting there. About 12 people have responded. I think for the sake of time, I'm just going to take a look at how you did. Again, you can always come back and look at these problems on your own if you'd like. Looks like kind of all over the place. Okay, so let's just briefly think about it before we sign off. If I had two monomers and I link them together, I get one molecule of water. If I take a third monomer and I link it to those two monomers that are already linked together, I get another molecule of water. Okay, so there's a general pattern that begins to emerge. For every new bond that you form, as you link these monomers together, you get out a molecule of water. So if I was going to link together 25 monomers, 25 monosaccharides in this example, I should get out 24 molecules of water, or n-1, where n equals the number of monomers that I'm trying to link together to build the larger polymer. Okay, so it looks like some people were, now your answers are changing, that's okay. Some people were spot on in terms of their 24, okay, but it certainly isn't going to be 50, and it's not going to be anything less than 24 either. Okay, all right. So again, lots of practice questions that you can look at. We'll circle back to these next week when we get the chance to meet again. And I think, for the sake of time, this seems like a good place to sign off. So I'll see you, my gosh, in 7 days. Please take care of yourselves. Take this time to take care of yourselves, take care of each other, and your family members, and be well. And I'll see you when I see you. Okay?
Thank you. Have a good break.
Thank
Thank
you.
you.
Love you. Bye.
Oh, you
Have
know.
a good break.
Thanks, guys.